Well, good morning for those in the room and those who are online. I appreciate your joining us for Elizabeth Oice's master's presentation this morning. So I'm really excited to introduce Elizabeth. She's been a student in our department since the summer of 2020 after she finished her undergraduate degree at Iowa State University. And while she was at Iowa State, she supported the research of Dr. Matt Liebman's team in the agronomy department, where she and I connected over a similar project to what she's going to share with you today, something I think is really interesting about weed seed banks and crop rotations. So Elizabeth is originally from a farming and rural community in Northfield, Minnesota. And I want to say it's really been such a pleasure to work with Elizabeth, and we do have a lot in common beyond both being Iowa State alum. She loves dogs, ice cream, sandhill cranes, and we're both really passionate about sustainable agriculture and the environment. But more important than all that, I'm really proud of a lot of things and happy to have worked with Elizabeth. She's really impressed me every time we meet that she is so sharp. She brings clever ideas to the table. She's got a strong grasp of the literature, practical agronomy knowledge, really strong communication skills. And I'm also just really proud of her resilience. You know, I say when she started because she's only been here and finished her undergrad all through the various phases that we've had of the pandemic, where we don't have, you know, our typical support and social systems in place. So I know it's been a challenge for her and a lot of students. I just want to recognize that. But in spite of it, she's maintained a strong community, many of whom are with us virtually and in person today. So with that, I'm going to turn it over to you, Elizabeth.
Okay. Thank you, Andrea. That was a really kind introduction. As mentioned, my name is Elizabeth Oice. I'm a master's student in the Resilient Cropping Systems Lab, advised by Dr. Beisch. And today I'm going to be discussing what I've been working on the last 2 years here as a part of my master's program, which is winter cover crop impacts on weed dynamics in eastern and central Nebraska, using cover crops to expand the integrated weed management toolbox. Today, if I can get this to work, I will be starting out with an outline, so an introduction to Nebraska's agronomic practices, herbicide dependence, and no-till row crop systems, as well as cover crops as a cultural weed suppressant. And then I'll be discussing the first and second chapters of my thesis, which are winter cover crop impacts on soil seed banks and winter cover crop impacts on weed growth during the growing season. And I'll be discussing objectives, materials, and methods, and results and conclusion for both, before wrapping up with a brief summary talking about weed management implications with cover crops and outcomes we've learned from both experiments. So as we know, we're located in the state of Nebraska, which is in the western region of the US Corn Belt. And currently the state has 22.2 million cropland acres in production. And the most important crops to the state are corn, soybeans, small grains, especially in the eastern part of the state where we have more moisture. As we move further west in the state, the land becomes more arid and we see more crops such as small grains, dry edible beans, and even potatoes. So in the eastern part of the state, our predominant rotation, because of that moisture factor, is a corn-soybean rotation, and occasionally extending that with a small grain crop. Furthermore, almost half of the cropland acres in production are under no tillage. If we think about what tillage is traditionally used for, it's not only to prepare a seed bed for the cash crop, but also to bury and destroy above ground weeds and weed seed. So if we reduce tillage or eliminate it altogether, we're limiting our weed control options to chemical and cultural methods. And we would define a cultural weed management method as any non-chemical practice that minimizes weed interferences with crops. So most commonly we see crop rotation, basic agronomics like row spacing. Today we're talking about cover crops, and also the timing of certain inputs would also be considered a cultural practice. For the most part, most growers do utilize some kind of strategic crop rotation if they're not limited to continuous corn. And we know that there are benefits to diversifying or extending crop rotations in that we can reduce weed pressure, but of course it is not a complete form of weed control. Instead, we depend upon herbicides for complete and effective weed management. And we can see that growers agree with this based on their management practices. So for example, 94% of all corn and 96% of all soybeans planted in the state are genetically engineered with stack tolerance traits of herbicide tolerance and insect tolerance. So common herbicide trait platforms you'll see on the market are Enlist E3, Roundup Ready crops, Liberty Link beans, and Extendiflex, for example. And herbicide tolerance is a great technology for us to use because we can apply specific herbicides as post-emergence herbicides without worrying about crop damage or death that would usually occur. And with the release of the Roundup Ready crops in the 1990s, glyphosate has risen to the most commonly used herbicide globally and in the state of Nebraska, followed by 2,4-D. As you can tell, this might be setting the stage for some problems in our cropping system, and one such problem is herbicide-resistant weeds. And so herbicide resistance comes about basically from the repeated selection pressures induced by herbicides that are applied year after year, similar modes or sites of action. And because of this, herbicide resistance is recognized as a global issue in the agriculture industry and is very problematic in the US Corn Belt as well. What herbicide resistance does for us is it narrows our options for viable herbicides. Or it can also cause us to increase our rates of those herbicides to maintain that level of efficacy. And if you think about the rising costs of inputs in 2022, that can get quite costly over time. Currently, Nebraska has 16 reports of herbicide resistance weeds, and we know that these aren't necessarily isolated cases and that these populations are somewhat widespread. And additionally, there's been 4 confirmed cases of multiple resistance, which means that these plants are resistant to multiple sites of herbicide action. These 4 cases all happen to be amaranthus species, which are known as pigweeds, such as the weed you'll see here in this slide. There are also other factors as well that impact herbicide efficacy, including climate change, and more specifically, the concentration of carbon dioxide in the atmosphere. And this may enable weeds to metabolize common herbicides such as glyphosate more efficiently, specifically with plants with a C4 photosynthetic pathway, such as pigweeds as well. Additionally, if we think about how long herbicides have been available to us, it's only since 1944 with the release of 2,4-D and 2,4,5-T. So that's really less than a lifetime that these products have been available. And additionally, there's been no new modes of action. There's been talk of it, but there's really been no new products out with modes of action for the last 30 years for herbicides. And so really the question is for us, how will herbicides work in the next 100 years, and what's herbicide resistance going to look like over time? So given this, there's a precedence for finding effective integrated weed management tools, also known as using multiple weed control tools in one weed management program, in order to maintain that level of efficacy over time, which is our goal with herbicides. However, the issue is getting growers and consultants to recognize the importance of integrated weed management. So for example, this is a survey done in the mid-southern United States in cotton, rice, and soybean systems. And they asked consultants to rank herbicide resistance best management practices in order of importance. And what you'll notice is that these integrated weed management tools or cultural methods are listed relatively low on the scale, and cover crops is actually listed second to last. And so what we'll see here is that the most important methods are actually directly related to herbicides themselves for herbicide resistance management. So there's kind of a disconnect for growers and consultants to recognize the value and importance for integrated weed management tools. So if we define cover crops more closely, we can define them as non-cash crops grown for the protection and enrichment of soil during periods that would otherwise be fallow or there's no crop growth. And traditionally, we recognize cover crops as soil health and soil erosion tools, but they can also provide weed suppression benefits, and they can do this through physical mechanisms. So to begin with, the living cover crop has direct competition for light, space, nutrients, and water resources, especially in that early growing season. So there's that interplant competition aspect that can help disrupt weed life cycles. We also have the living cover crop and its residue can lower the quality and quantity of light into the soil profile. And the shading effect can actually change the soil microenvironment by reducing soil temperature over time. We know that those last 2 factors can directly impact weed seed germination or delay weed emergence. There are also chemical mechanisms for weed suppression under cover crop, and one such method is with allelopathy, which is known as the plant's ability to excrete chemicals into the soil environment around them or contain those compounds within their residues, and these act as germination or growth inhibitors to nearby weeds. Furthermore, there's been evidence that there's synergism with herbicides and cover crop residue together, meaning that we can secure better weed control with both those methods compared to using one or the other alone. Also, it is important to recognize that we typically secure better weed control with greater amounts of cover crop biomass. So greater cover crop biomass can typically lead to reductions in weed biomass and weed emergence. And if we talk about the latter, weed emergence, we're pretty much directly talking about the weed seed bank, and we can define that as a reserve of viable weed seeds on or within the soil profile. And weed seed banks are excellent for providing insights on weed reproduction, species population dynamics, but more importantly, the potential for future weed infestation based on the number of weed seeds in the soil or the seed bank density. Furthermore, seed banks aren't a year-to-year thing. So seeds can actually stay viable in the soil for long periods of time. And depending on what literature you look at, for example, pigweeds can stay present in the soil anywhere from 12 months to up to 40 years. So that's quite a long time to have a viable seed bank. And I kind of want you to think about a weed seed bank as a bank account, right? So we have withdrawals and deposits. Withdrawals in the seed bank can look like germination, it could be seed death or failed germination even. And deposits are just the return of weed seeds back into the soil. And so if we have more withdrawals than deposits, as the schematic shows, then over time we might expect that bank account balance to shrink. So if we apply these to a couple of different hypotheses out in the field, let's say we have a field that's under no tillage production and it's herbicide managed, and we establish two different plots with the same starting soil seed bank density. If we apply a cover crop treatment to the plot on the left, and we apply what we've learned so far about the physical and chemical impedances on weed seed germination, we might expect that under the cover crops, there would be reduced withdrawals through weed germination compared to the non-cover cropped plots where there are no physical or chemical impedances to weed seed germination, and those seedlings can freely emerge. Additionally, since this is a herbicide controlled plot, we're going to assume for simplicity that there are the same rates of deposit of weed seeds back into the soil seed bank, since typically herbicides would take care of any other emerged weeds. The only other difference here would be some enhanced withdrawals through predation and natural seed death with the soil seed bank and the cover crop. And over time, if we looked back at the soil seed bank density in a couple of years, we might expect that the cover crop soil seed bank is larger in comparison to the check because of those reduced germination withdrawals. Another scenario or hypothesis we could apply in the field is still continuing reduced withdrawals through weed seed germination, but instead we have enhanced withdrawals through weed seed predation and natural seed death. And there's been evidence that cover crops can provide really excellent habitat for weed seed predators, including invertebrates or even mammals. And those moisture conditions that cover crops can induce can enhance natural seed death or decay over time. So alternative scenario is if we looked at the seed bank many years later, we might see that that soil seed bank is actually shrunk quite a bit. So those are two different options or hypotheses that we have for out in the field with soil seed banks under cover crops. So given these physical and chemical suppression mechanisms of cover crops, our primary thesis objective was to determine the efficacy of winter cover crops as a cultural weed suppressant and quantify those impacts on both weed seeds and growing weeds in herbicide managed cropping systems. This led us to design two separate experiments. So the first chapter being the below ground soil seed bank experiment, and the second being the above ground weed growth experiment. So both of these give us the two sides of weed dynamics out in the field. Jumping into the first chapter of my thesis, our primary objective was to sample and germinate the soil seed bank to determine if seed bank size and species composition differ between cover crop and non-cover crop plots. We utilized 6 different sites across eastern and central Nebraska. 4 of the sites were on-farm research collaborations with the NRCS Soil Health Initiative. And these growers set up these experiments with the intention of understanding the impacts of cover crops on soil health and yield initially. And these were located in Colfax, Greeley, Howard, and Merritt counties. Additionally, we utilized 2 UNL research stations with USDA cover crop plots at the Eastern Nebraska Research and Extension Center, which I'll call NREC, and the South Central Agriculture Laboratory, which I'll call SCAL, and those are labeled in yellow. The on-farm sites were established in 2016 or 2017 and had a total of 4 to 5 years of cover crops present up until our time of sampling. They included 2 different treatments, which was a multi-species cover crop mix ranging from 8 to 12 species, depending on the site, and a non-cover cropped control, which I'll refer to as a check. 3 of the 4 sites were under no tillage production, and historically these sites were corn soybean rotations, but in 2020, the year prior to our sampling efforts, the rotation was diversified with a small grain or cool season legume. And then in 2021, the year of our sampling, 3 of the 4 sites grew corn and 1 grew soybean. SCAL and NREC, these sites were established earlier in 2014 for a total of 7 years of cover cropping. And the treatments included cereal rye, hairy vetch, or the non-cover crop check. And for the duration of that experiment, it was a corn-soybean rotation, and they were also under no tillage management. Just to reiterate, the on-farm locations are full field scale experiments. And so these plots ran the entire length of the field in production. So the average plot size across the 4 sites was 48 by 570 meters, whereas the research station sites were only 6 by 9 meters. And so this image here also gives you a good insight as to what those treatments look like. So on the left-hand side, we see the multi-species cover crop mix, even with the sunflowers from the fall previous. And then on the right-hand side, that is the non-cover crop check that we'll be referring to. So in order to sample the soil seed bank, we went out in early April before the herbicidal cover crop termination occurred. And we took 20 random subsamples per plot to a depth of 10 centimeters. So within no tillage systems, the majority of weed seeds actually fall within the first 3 centimeters roughly of the soil profile. So 10 centimeters is quite a generous depth to obtain the viable soil seed bank. After we collected those subsamples, we mixed them together to form one composite sample per plot. And additionally, to account for plot size differences and the spatial variability at the on-farm locations, we accumulated 5 times more soil volume at those on-farm sites. After we brought that soil back to the university, we sieved it to remove any living plant matter or insects and also to break up soil clods. And then we placed them into trays, as you can see here, where we watered and monitored them daily. And those seedlings were able to freely emerge, and any seedlings that emerged were identified to species rank. And as you might expect over time, those germination rates slowed down. And in order to stimulate additional germination, we dried out the trays for 5 days, re-sieved them, and then repeated that watering process. So there was 3 rounds of germination that occurred with the re-sieving in July and September. And the experiment ran a total of 7 months between April and November. Any of the weed seeds that were counted were converted into estimates of weed seeds per meter squared, which is known as our seed bank density. And the species identified were categorized by functional groups of pigweeds, grasses, other broadleaves, and the sum of those 3 categories make up the total. We analyzed the data utilizing generalized linear mixed effect models with a log-linked negative binomial distribution. And we modeled this with the LME4 package in R, and we utilized a significance level of 0.1. Additionally, we utilize non-metric multidimensional scaling, or NMDS, which is a fancy way of saying we plotted points to observe visual patterns in this complex and dense seed bank composition data set. We utilized the vegan package in R, and points were plotted with Bray-Curtis distances, which is a common dissimilarity index used in ecological data. Because we were also interested in understanding how seed bank composition and diversity change under cover crops, we looked at 3 different diversity metrics. So this included species richness, which is defined as the total number of species present in a community, Shannon diversity, which takes into account the proportional abundances of all species, and then Pylus evenness, which accounts for both diversity and richness and scores 0 to 1, 1 meaning that we have a completely even population, and all those species that are found in the same amounts. Additionally, because of experimental design, treatment, and spatial differences, the on-farm and research station models and analyses were run separately. This is kind of what the process looked like in the greenhouse. So for example, identifying a grass ligule, a grass seedling based on its ligule. And also what you might notice is that certain trays are dominated by grass seedlings, others dominated by pigweeds, or maybe a mix of both. And there's great variability even between the treatments and the sites. And some of the most common species we found, well, the most common species we found was Palmer amaranth, which can be identified based on those water markings on the leaves and also the long petioles. Other weeds, common weeds such as shepherd's purse and green carpet weed were also found. And native plants, including Western tansy mustard, which was one of my favorite weeds actually, was also found in this mix. So quite a diversity of species found. And across the 6 sites, we germinated over 6,500 seedlings composed of 57 different species. Palmer amaranth was the most commonly found species across the 6 sites, making up just over 1/5 of the total seed bank. And then that was followed by green foxtail being the 2nd most common species. Other pigweed species such as red root pigweed and common water hemp were also other commonly found species. And for the most part, we observed summer annuals. And in that second flush of weeds, that germination round, we observed more winter annuals emerging. You'll also notice that we're utilizing a species code, which is a unique identifier code representing the first 3 letters of the genus and the first 2 of the species. And this unique identifier code will become more important when we look at the NMDS analysis in just a minute. When we looked at total seed bank density, we saw no difference between the treatments at any site. On the low end, the seed bank density ranged from 165 seeds per meter squared up to over 4,000 seeds per meter squared at the Merritt County cover crop site. However, when we started to notice differences was when we looked at the pigweed portion of the seed bank. So we observed increases in the cover crop at 3 sites and 4 treatments, representing 180, 137, 243, up to 355% increases compared to the check. If you refer to the last slide as well, the pigweeds make up most of the total seed banks at each site. We also utilize non-metric multidimensional scaling, as I mentioned, and I don't want you to be overwhelmed by this graph. In fact, you can omit the values on the X and Y axes, and rather what I want you to pay attention to is where these species codes are plotted. And at each site, they're plotted based on their relative abundance of one another. So species, for example, that are circled there are found in relatively the same amounts across that site, whereas species plotted oppositely are found in very different abundances. Furthermore, you'll notice colored points on each of the graphs, which represent 1 plot per treatment. And those are plotted based on the average number of species found, but also their relative abundance. And we overlapped these points with a polygon that can help represent the bounds of that seed bank's composition, the species composition. Now, if we notice any overlap between the polygons or even within the plots themselves, this means that species composition is very similar. However, when we start to look at Greeley and Merritt County in specific, that's where we start to see some differences. And so we see complete separation of both of the treatments, indicating that cover crops have a unique species composition. So remember, this is the same site, same field. And so if we're assuming that the check and cover crop treatments started out with the same seed bank composition and density, this means that cover crops are likely inducing conditions that shift species composition over time. Furthermore, as I mentioned, we utilized 3 different diversity metrics, and we computed an estimated change value for each, which represents the average check treatment score minus the average cover crop treatment score. And so essentially, when you're seeing a negative value, that means that the diversity metric improved in the cover crop treatment. So what you'll notice is we didn't find any statistically significant results, except for actually a reduction in evenness at the Merritt County site, which makes sense because we saw dominance of pigweed species in the cover crop there. However, what's important to note is that we observed numerical improvements in species richness. So the total number of species found in the cover crop was greater at all 6 sites and both treatments at scale. And species evenness also improved, except for at the Merritt County site there. Our primary conclusions from the first chapter are that cover crops influence seed bank composition, but they did not influence total seed bank density. So we saw that cover crops at 2 sites created distinct seed bank species composition at Greeley and Merritt County. But more importantly, we saw that pigweed seed bank densities were greater under cover crops at 3 sites and 4 treatments, up to 355% increases. So this is likely indicative of the reduced seed germination withdrawals over time that we talked about with seed bank scenario 1 in the introduction. Furthermore, while we did not see statistically significant results with the diversity metrics, there's still value in improving weed richness and weed evenness. While growers would initially perceive more species present in their fields as a risk, there are actually lots of benefits to diverse weed communities, including less interference with the cash crop, which has yield benefits. And you're also enabling the ability to diversify your mode or site of action over time with more diverse weed communities. If we think about this in an agroecological sense, these diversity metrics are useful indicators for the success of rediversifying systems. So for example, integrating cover crops into this agroecosystem. And this is a central component of making agriculture and weed control more sustainable over time. Furthermore, we recognize that our results may be due to the length of our experiment. So while 4 to 7 years is subjectively a long time to have cover crops, we would see a shift towards seed bank scenario 2 with longer term experiments. So for example, A study done in Iowa that had cover crops present for at least 10 years found that cover crops have the potential to reduce seed banks, and they found this at a few of their sites that included on-farm research. So with longer-term experiments, cover crops present for longer than 10 years, we may have more enhanced withdrawals through weed seed predation and seed decay. If we jump into the second chapter of my thesis, the primary objective was to quantify weed density and weed biomass to determine if cover crops interfere with weed emergence and weed growth during 2 points in a single growing season. Utilizing the same 6 sites as I introduced with the first chapter, we went out and obtained cover crop biomass before the herbicidal termination of the cover crop, roughly in mid-April at all sites. We converted these samples into estimates of megagrams of dry plant matter per hectare. And these images kind of give you an idea of what the process looked like. So on the left-hand side, we see at the Merritt County site, really excellent growth of the brassicas. And then on the right-hand side, that's kind of what the process looked like of obtaining the cover crop biomass in those set quadrats. And obviously some of us thought it was a little colder than others last spring. As far as above ground weed growth samples go, we obtained weed density and weed biomass samples twice during the 2021 growing season. We first went out in the early growing season, pretty much immediately after that cash crop emergence, but before post-emergence herbicides were applied. This time frame fell within late May and early June. And we chose this time period because it falls within the critical weed control period, which is basically known as the time frame where weeds have the greatest potential to interfere with crop yield. Additionally, we went out during the late growing season, 3 to 4 weeks after a post-emergence herbicide was applied to essentially capture that second flush of weeds, but before canopy closure, which was roughly V10 vegetative stage in corn. Additionally, this fell within the late June and early July timeframe, and we wanted to know if cover crops are having an impact on weed density and weed biomass. Do these effects persist while that cover crop is degrading out in the field? We took these samples 3 times per plot using spatially randomized samples, spatially randomized points plotted with ArcMap, and these points were GPS marked and flagged in the field in order to return to the same locations for the late season samples. It's also important to note that we took weed density and weed biomass samples in separate quadrats so as not to disrupt any density counts. And likewise with the soil seed bank experiment, we categorized weeds by functional groups of pigweeds, grasses, other broadleaves, and totals. So our weed density, so the number of weeds that emerged, were converted into estimates of weeds per meter squared. And weed biomass, so the size of weeds essentially, was converted into estimations of grams of dry plant matter per meter squared. We log+1 transformed the data set in a linear mixed effect model, which was also modeled with the LME fork package in R and utilized a p-value of 0.1. I will also say that it's important to note that we treated Colfax County essentially as an outlier because we simply found no weeds at any of these sample points in any treatment or any plots. So it is omitted from the graphs today. We saw that on the low end of cover crop biomass, it ranged from 0.1 megagrams per hectare to just over 4 megagrams per hectare at the Merritt County site. You'll also notice that the multi-species cover crop mixes accumulated more total biomass than the singular species at Scal and NREC. In part, this is due to an earlier plant date at these on-farm sites because the cover crops were planted following a small grain or cool season legume harvest. So if we look more closely at these planting dates, we'll see that these cover crops at the on-farm sites were planted really early, even late July and early August for those multi-species mixes versus NREC and Scal were planted in October and November, but all sites were roughly terminated in the same time frame in the spring. When we looked at total weed density, we saw reductions at NREC in the late season samples and Greeley County during both sampling periods, which represented 73, 87, and 89% reductions compared to the check. It's also important to note that there were small amounts of weed growth in general in most plots at all sites, and then that these reductions, at least at Greeley County, were driven by reductions in emerged grasses. You'll also notice that at the research stations, the overall weed density was greater than the on-farm sites, and we can attribute this to a couple different reasons. So firstly, if we looked more closely at the herbicide programs, we would see that there are no residuals or soil activity in the pre-emergence herbicides at Scal and NREC, and also in the post-emergence at NREC as well. Furthermore, if we think about the cropping system or the crop rotation history of these sites, the two research stations have been corn, soybean the entirety of the experiment, whereas the on-farm locations had a diversified location where sampling the year after a small grain harvest. What's interesting though is when we look at pigweed density, and what we saw is that despite these major increases in the pigweed seed bank, there were no differences in above ground pigweed density. So this means that even though there are large amounts of pigweeds present in those cover crop plots in the soil, they are simply not being expressed above ground. If we look at weed biomass, we also saw reductions at two sites, and so that represented 96, a 99, and an 88% reduction compared to the check. Likewise, at Greeley County for the weed density, the weed biomass reductions were driven by grasses at both of the sites and also broadleafs at Greeley. What's a little different about weed biomass here is that we observed consistent numerical reductions at all sites and sampling periods, whereas we did not observe this with total weed density. Furthermore, we had relatively small weed biomass samples, and so all of the weeds that we sampled were typically less than 3 centimeters or 1 inch in height, so very small. And additionally, with pigweed biomass, the only reduction we noticed was a reduction at NREC in the late season samples, representing a 93% reduction compared to the check. Our primary conclusions from this chapter were that cover crops significantly reduced weed biomass and weed density at some sites and sampling periods, but this effect was not consistent across all sites. We recognize that obtaining more cover crop biomass may lead to more consistent results, but in general, there's a challenge for obtaining this amount of cover crop biomass in the Corn Belt due to cropping rotation limitations. So, if we're following a corn or soybean harvest, if we're lucky and we're drilling cover crops, really the earliest we could get in there is the end of September if we're really lucky, or October. And then we're still terminating at the end of April, early May. So that time frame to accumulate growing degree days and accumulate above ground biomass is really challenging. And it's also important to know that these on-farm sites planted cover crops really early and still did not reach this level of biomass. However, we still observe reductions in both weed biomass and weed density at several sites, even without this optimal level of cover crop biomass. So there's an importance of interplant competition that's likely at play as well. Furthermore, we must take the results in context of the management system. So all of these sites were managed with pre and post-emergence herbicides. And typically with weed science experiments, we'll utilize some kind of herbicide-free control, both with a non-cover crop and a cover crop plot to remove that confounding effect of herbicides. However, when we're working with on-farm and real-life growers, our results are a little bit more practical because these growers rely on herbicides every growing season. And our results are a little bit more applicable because growers just want to find out how cover crops would react in their normal management system. More importantly, though, we noticed that above ground pigweed densities were unchanged in cover crops despite these major increases in pigweed seed banks. Additionally, this pigweed suppression was still likely occurring without large amounts of cover crop biomass at those optimal levels. And again, this reduced withdrawals through weed germination is indicative of that seed bank scenario 1 that we discussed earlier. So this may be beneficial to both pigweed and herbicide resistance management. So if we refer back to seed bank scenario 1, again, remember we expected that there would be reduced withdrawals through weed seed germination for the total seed bank. And while this did not have happened for the total seed bank, what we found instead was this was happening for the pigweed portion of the seed bank. And we confirmed this both with seed bank size and the weed above ground pigweed density. So our primary conclusions from both of these chapters were that cover crops suppress pigweed seeds or these amaranthus species through reduced germination withdrawals. So even if pigweed seed banks increased all the way up to 355%, they were not expressed above ground in cover crop treatments. Again, this is indicative of seed bank scenario 1, and these suppression benefits were still occurring without optimal levels of cover crop biomass. Therefore, we conclude that cover crops may be a viable tool for pigweed seed bank management and ultimately mitigating herbicide resistance risks. So if we keep weed seeds ungerminated and dormant in the soil seed bank, ultimately what we're doing is keeping more weeds unexposed to selection pressures that drive herbicide resistance and those related mutations. We're also limiting our contributions of weed seed deposits back into the soil. So given our results and in context of these seed banks, we believe that herbicide resistant management strategies should be reframed to include not just above ground weed growth, but also seed bank management in general. For future research, we believe that it would be beneficial to quantify the seed bank before the establishment of a cover crop experiment. This way we could better track the trajectory of seed banks over time, and it would also give us insights on weed seed persistence over time, especially with those pigweeds. Furthermore, it may be wise to separate the effects of crop rotation, since that has known effects on above ground weed density as well as seed banks. And in an ideal world, we would have liked to keep herbicide programs consistent across sites, but that is one of the many challenges that we face utilizing on-farm research. So that being said, I have really enjoyed my time here at UNL these last 2 years. And even though I did come into the pandemic, I've had a lot of great opportunities, both within this department and the community. And so I have had some opportunities with extension and outreach. And so last summer, alongside Dr. Kohler-Cole and Chris Annual, we presented in the cropping systems lectures, and I got to present some preliminary results for the projects I've just discussed here with you today. And so I love doing that and getting some face-to-face time with growers. And if you haven't already seen my team here, we've recently published a Crop Watch article talking about these same results here in more of a synthesized version for growers on Crop Watch. So I would also like to take some time to recognize a lot of important people who have made this experience and research possible. I would first and foremost like to thank my advisor, Dr. Beisch, for all of her mentorship and support these last 2 years. And then my committee, Dr. Kohler-Cole and Dr. Proctor, for their insights and help on my thesis and some professional development opportunities. And then in general, I have really enjoyed working with the Resilient Cropping Systems research team as a whole. Not only has their help in the field been wonderful, but the general morale of this team and a primarily female-based team too has been absolutely wonderful. And I thank Tom as well for his support at NREC and also the undergraduate research assistants who have participated in the field, even when it was unpleasantly hot or cold. And I'd also like to give a shout out to the NRCS Soil Health Initiative team, especially Aaron Hurd and his team for their help with the field work in the spring of 2021 and also the general morale of doing soil health and on-farm research. And I'd also like to thank the grower collaborators at the Colfax, Merrick, Greeley, and Howard County sites for offering up not only their land, but their time and also having curiosity for on-farm research and cover crops in general. And of course, this goes without being said. I thank all of my family and friends who are present on Zoom today or in this room here too. So thank you for being a constant sounding board and having genuine or feigning your interest in my rants about this research for the last 2 years. So with that being said, I want to thank everyone for their time and attention today. And I would be happy to answer any questions now, but if you also think of any questions later or have any curiosities, feel free to send me an email. But yeah, I'll open up the floor for questions now.
I've got a couple of logistical questions. One, I wasn't clear, maybe I just didn't catch it. Very nice presentation, by the way. How and when, you gave when you terminated the cover crop, how was it terminated? And would that have an impact perhaps on your future research?
Yes, so all of these sites were terminated with herbicides, and I can also get up the herbicide program and specific ingredients if you're liking as well. So yeah, all these sites were sprayed roughly late April, early May. And so, of course, these results are in context of herbicides. So the above ground weeds certainly likely had an impact, especially if they had the pre-emergence herbicides had some kind of residual in them. But does your question pertain to the soil seed bank or the above ground weeds, I guess, too?
And the other question, you are from Minnesota, correct?
Yes.
And in one of your first slides, you showed relative use of cover crops. And Minnesota seemed to be way behind in the gray region instead of the pink or purple or whatever the high ones were. And I wondered if you had a rationale for why that might be the case.
But that was actually probably for no-till acres, but it probably follows the same trend with cover crops as well. And yeah, there is a rationale. Having worked up there before, the primary consideration is soil temperature, actually, because a lot of and this occurs with no-till too, right? Same factor. If we're tilling and we can get the soil warm enough to plant corn, at least in the early spring, that's what most growers are focused on. And also just that time frame to accumulate biomass and the growing degrees days it takes to get enough cover crop biomass to have some kind of soil health or weed effect. It's quite limited, especially if you move up north in northern Minnesota. Those growing regions vastly differ from where I'm at in South Central up to, closer to North Dakota. So there's some just timing logistics with that, but also agronomic concerns regarding mainly soil temperature.
Thanks.
I'm aware that we have some questions on the chat. Does anyone else want to ask a question here in person?
All right, great. The first question online is from Stefan. He says, thank you for this wonderful work. What do you think would happen if the cover crop strips from all your sites reverted to no cover? Could that result in a nasty pigweed problem?
Yeah, definitely. Right, because there's no physical or chemical impedances. So those seedlings probably could emerge freely and you would probably have higher rate of infestation, especially if, let's say, you're managing organically, we'd have some major issues there as well. So yeah, I guess that would be a trade-off if you decided to eliminate cover crops at some point, especially in those specific plots.
Great. Okay, Gina has a question online. She says, great presentation, Elizabeth. I'm curious if you have any specific ideas what might be preventing the pigweed seeds from germinating in the cover crop treatments?
So it's interesting. I'm trying to wrap in my head around that too over time, that it's not the total seed bank density that's affected, it's just that pigweed portion it appears. So I'm not sure what exactly is impacting this. It could be that pigweed seeds they're smaller seeded species, so the amount of light and the depth at which they're buried could impact their germination, but also perhaps allelopathy and those chemical factors maybe playing a role in that. But yeah, it is interesting that it's just those pigweeds in this case that are being impacted.
Great. Okay, we have another question online from David Meyer. David says, nice job, good study and presentation. This may not be a fair question, but can you speculate on what your results might have been if used in an organic system with no herbicide use?
Well, we would probably, let me think about this. So the herbicides are definitely playing a role with our above ground results too, in both of the treatments. I would say that if let's say you didn't have great cover crops the next year and you're in an organically managed system, you probably would have some sort of pigweed infestation going on without those physical or chemical mechanisms for germination to stop. So I would say if we're seeing the seed bank scenario 1 where we're having those reduced withdrawals, it probably would be more problematic over time with organic systems compared to that second scenario where we're talking about those enhanced withdrawals through predation and seed decay. So I guess you probably want to see seed bank scenario 2 if you were using some kind of organic system without herbicides. So yeah, there's definitely trade-offs.
Do we have any other questions in person? Okay, one last in-person question.
Thanks, Elizabeth. This is really cool and very great presentation too. I just have a question out of curiosity. I know that wasn't anything that you necessarily recorded or have in the thesis, but when you did the seed bank, when you germinated, you said you had, I think, 3 flushes of. Did you notice different species emerging over time, or was it pretty much always the same species kind of?
So I would say I kind of alluded to this, but that first flush, that initial flush straight from the field, that was primarily summer annuals. And then what was interesting was that with that second flush, so after we dried out the trays and resieved, that's when I started to notice quite a few more winter annuals emerging too. So yeah, and so yeah, and then another way we could have gotten more winter annuals, I guess, to emerge is if we did some kind of cold stratification with the soil to break dormancy cues, but that would have been probably another year onto this experiment. So yeah.
So with no more questions on the chat or in person, I suppose that concludes Elizabeth Lloyd's defense.
Gina asked, actually, what's next for you, Elizabeth?
Things are in the works. So I'm actually hoping to return back out to industry and working in technical agronomy or sustainable agriculture. So
In Minnesota.
in Minnesota,
So
hopefully.
someone
Yeah. Yeah. No, thank you everybody for attending online. I really appreciate it, your time and attention.